First of all, I would like to thank the organizers for inviting also Second Genome. And today I'm going to talk about the microbiome in complex diseases. Just as an introduction, what we understand for microbiota and microbiome. Microbiota is an ecological community of commensal symbiotic and pathogenic microorganisms that we find in and on multicellular organisms. And microbiota include bacteria, archaea, protists, fungi, and viruses. Microbiome refers to the combined genetic material from all the microbiota. And just here, oops, sorry. Just here, you can see the picture that represents the amount of combined genetic material from the microbiota, or in this case, the gut microbiome, compared to the human genetic material, in this case, the host that is the human, is overwhelmingly high compared to the human. And just a few highlights on the human microbiota. We are germ-free at birth. Basically, we don't have any microbiota on us. We are colonized during postnatal period. And by adulthood, our bodies become home of trillions of microbes, which outnumber our own cells, an estimate of like 10-folds. Then this is a huge amount of genetic material. The Human Microbiome Project has described several habitats for the human microbiome. In the mouth, pharyngeal, and respiratory system, it has been estimated to have or to be colonized by more than 600 species. The skin, that is the most populated in number of species tissue in the body. It has been estimated more than 1,000 species, and it makes sense since it's the tissue that actually protects us from the environment. The urogenital tract and the stomach contain far less number of species, and the intestines that today I'm going to focus contain an estimated number of 500 to 1,000 species. And in the intestinal microbiome, we can hear like day-to-day species like Lactobacillus casei that we can see in the yogurts, and we are very close to these bacteria that we can hear on the grocery store or on the TV, that they are commonly found in the food. In the human intestinal microbiome, in the ileum and colon are the most densely colonized body habitats. And the bacteria that are the dominant by biomass are followed by archaea, eukarya, and viruses. And within bacteria, we have two phyla that dominate in mammals, the Bacteroidetes and Firmicutes. But what are this microbiome? Which are the functions of this microbiome? Microbiome has, let's say, high number of biological functions that have been described, and in multiple tissues. For example, microbiome is important in metabolism since it generates nutrients from diverse substrates, generates short-chain fatty acids, is involved in the synthesis of vitamin and transformed bile acids. And it also helps to metabolize drugs and xenobiotics. Locally, the intestinal microbiome also plays important roles in the intestinal functions, such as inhibition of new colonizers, and produces antibacterial products. It also is also involved in the maturation of the epithelial barrier, as well as the intestinal motility. The microbiome also has been associated to biological functions outside the local area where it's present, or systemically. For example, in the central nervous system, it plays a role in the synthesis of neurotransmitters, is involved in hippocampal neurogenesis, cortical myelination, and blood-brain barrier function, as well as in inflammation. And obviously, the microbiome is playing a very important role in the immune system, maturation and homeostasis. It plays a role in antibody production, and it has been observed that germ-free mice that do not contain microbiota are more susceptible to infections. The microbiome has been also associated to other biological functions, such as vascularization, bone density, or cardiovascular functions, between others. Any change in the microbiome resulting in a microbial imbalance also called dysbiosis, has been associated to a large number of complex diseases. For example, atherosclerosis, inflammatory bowel disease, including ulcerative colitis and Crohn's disease, nutritional disorders such as bulimia and anorexia nervosa, neurological disorders, including Alzheimer's disease, Parkinson's, and autism, autoimmune disorders, such as rheumatoid arthritis, between other autoimmune disease, and more recent articles are showing that the microbiome is also playing a role in anti-tumor immunity. Today I'm going to focus on two important aspects, or mostly the classical aspects of the relation between the microbiome and the disease that is type 2 diabetes and obesity. And there are two main reasons why Second Genome is interested in working on type 2 diabetes. One is because, year after year, the increased number of patients that are diagnosed with type 2 diabetes is increasing. And data from the World Health Organization describe that we have more than 422 million people worldwide that are diagnosed with diabetes. 2.2 million deaths are consequences of, these patients die from consequences from having high blood sugar, and 50% of them die of cardiovascular diseases. 2% from 10% people develop visual impairment or completely blindness. 10 to 20% die of kidney failure, and the risk of dying is twofold compared to their peers. Therefore, this is a worldwide epidemic that we would like to contribute in possibly stopping it. And the other reason is that initial work on the microbiome and health, human health, was actually showing initial correlations between metabolic diseases and the microbiome. Later studies start identifying the bacteria and their functions that are important in metabolic functions. At the same time, they start associating the gut microbiota between healthy individuals and type 2 diabetic patients. Later studies, they start showing the role of the microbiome in improving metabolic functions. And more recent papers have started to demonstrate the initial, the causality in mice and humans. For example, these two articles are showing that from twins with different, one with metabolic syndrome and the other one lean or healthy, when we transfer the stool samples to germ-free mice, the mice that receive the stool samples from the, let's say, the obese twin or the type 2 diabetic twin, the mice became obese. However, the mice that received the stool sample from the lean patient or lean twin did not suffer any change metabolically. Also, other human studies show that stool samples transferred to patients with metabolic syndrome improve their metabolic outcome. And more recently, some other publications have started showing some that specific microbial species have some beneficial metabolic outcomes. For example, in these two papers where Akkermansia mucinifila or Faecobacterium prausnitzii have shown some improvements at the level of insulin resistance and liver disease. These are the reasons why, while others are working on prebiotics, consortia, and probiotics, at Second Genome we are interested in identifying the specific bioactives that, as well as the possibility to generate to activate or inhibit mediated by generating agonists or antagonists against these biofactors. Although we are primarily interested in these bioactives, bioactive meaning by proteins, peptides, and other molecules, including metabolites, we are also interested in identifying the specific strains that actually generate these bioactives. And this is key because the intestinal microbiota is extremely complex. and diverse. This is just an example of the same genus, Bacillus, containing hundreds to thousands of different species. And even inside the same species, we can observe that there is the presence of like hundreds to thousands of different strains. Then that's one of the reasons why it's so complicated, but at the same time it's key to identify which are the specific strains that are generating these bioactives. For example, just at the level of this Bacillus genus, we observe that we have the Bacillus anthracis that causes the anthrax, and we have Bacillus cereus that is a common probiotic used if you go to any grocery store, you will find Bacillus cereus and you can just buy it and take it and it's healthy and doesn't happen anything. At Second Genome, we have developed a platform by which from human microbiome analysis as well as strain-specific analysis, we're able to identify specific bacterial strains as well as the bacterially derived therapeutic bioactives. With these bacterially derived bioactive, we screen them through a series of experiments that will help us to elucidate whether these bioactives are beneficial or not for immune regulation and metabolic diseases that I'm going to talk today. Which human samples we have been using for generating some bioactives that could have potentially beneficial aspects for metabolic diseases. We decided to use basically patients with metabolic syndrome, pre-surgery from RYGB and post-surgery of RYGB, drugs and wide gastric bypass surgery. And the reasons for that is that bariatric surgery is the most effective long-term treatment for type 2 diabetes. Most of the patients that go through bariatric surgery restore glycemic control, normalizing the HbA1c levels, eliminate the need for medication in most of the cases, and is a sustained effect. It has been observed more than 15 years. Then these patients don't need to medicate ever again. Prevents the disease progression and reduces the overall mortality. Then by using this the analysis of these stool samples from these patients, we were able to identify specific bacterial strains as well as their bacterially derived biofactors, and we are screening them for if these bioactives have any beneficial effect. And the support for that also is that there is a strong correlation between gastric bypass and microbiome. There are two very interesting articles that show that the transfer of gut microbiome from RYGB-treated mice, as well as transplantation of human stools from RYGB patients, actually, when we transfer these stool samples to an obese mice, we are able to restore the metabolic control on these mice. Which is the screening approach that we have decided to take. It's a combination of ex vivo and in vitro assays in which we include liver explants, intestinal explants, as well as in vitro adipocytes and beta pancreatic cells, in this case MIN6. And we are going to analyze important biological functions that are affected in type 2 diabetes. For example, insulin sensitivity, gluconeogenesis, lipolysis, insulin production, and the production of gut hormones that have shown beneficial aspects in metabolic syndrome. And this is a decision based on the complexity and multiple tissues that are involved or that are affected in type 2 diabetic patients. We have decided to analyze key molecules in the insulin signaling pathway, as well as the end result of these signaling pathways that are the biological functions. And I'm just going to show you some few data that we have obtained that by using the liver explants and treating these precision cut liver slices with a cocktail that increases gluconeogenesis, basically glucose production from these livers, we're able to shut down by treating these liver explants with some specific peptides that we nominated through this selection process from these patients. And you can see that they are, in this case, these two peptides, basically that's the control, the blue and the green ones are the cocktail treated mice, and then liver explants from mice. And the blue bars are the peptides that did not have an effect. However, we observed 2 in this case that had a strong effect, even stronger than metformin, that is the primary therapy used for type 2 diabetic patients, but mostly for hyperglycemic, to restore the glucose homeostasis in these patients. Then, just wrapping up, the story is that microbiome plays an essential role in maintaining human health. Dysbiosis in microbiota leads to complex diseases. Microbiome has the potential to improve complex diseases. And we identifying specific strains and bioactives with beneficial effects is key to develop therapeutic treatments. And what we are doing at Second Genome to basically go from the microbiome to a medicine. Then we are mining the microbiome for novel therapeutics, and we are focused beyond the concept of Baeza's drugs. We are interested in the specific strains and the specific bioactives from those strains. We have a broad and leading microbiome bioinformatics platform. We are able to identify therapeutic pathways, molecules, and targets, and we have the capability to express and screen microbiome bioactives. And right now, at this moment, we have a therapeutic pipeline serving large unmet needs, and we have a phase 1 drug and additional lead compounds, and we have a very strong preclinical and discovery programs in gastrointestinal, metabolic diseases that I've shown you slightly today, and cancer immunotherapy. And just as a final thought, it has been actually also like almost from 70 years that we have been avoiding the presence of bacteria and fungi and viruses, cleaning ourselves and treating with antibiotics. And now there's kind of a revolution like that's changing that. We are more permeable, that we believe, to all these agents. And we are part maybe of a supraorganisms that we have to start like thinking about, like it's not only our health, but it's maybe also the health of the microorganisms that we have inside us that can make a big difference in that concept. And I'll just leave it here, and thank you very much for your attention.